In early May 2026, an infectious cluster emerged in one of the most isolated maritime environments on Earth. Passengers aboard a vessel traversing the South Atlantic began displaying signs of rapid, severe respiratory failure. The vessel was the MV Hondias, an expedition ship operating far from the reach of traditional medical infrastructure or emergency response teams. The World Health Organization identified the pathogen as Andes OrthoHantavirus. Their epidemiological data confirmed a case fatality rate of approximately 38%. While most hantaviruses are strictly zoonotic, the Andes strain is uniquely dangerous. It is the only known hantavirus with documented human-to-human transmission. Standard clinical interventions fail once the infection progresses. Nucleoside analogs like ribavirin show no efficacy in rescuing patients already experiencing the severe cardiopulmonary phase of the disease. This therapeutic failure is rooted in the virus's structural architecture. To a 20th-century virologist, a hantavirus was a strict and enclosed definition. It was a non-arthropod-borne pathogen, meaning it did not rely on insect vectors, that caused specific hemorrhagic and pulmonary diseases in humans. The global pursuit of these viruses began in earnest during the Korean War, when hundreds of United Nations troops were hospitalized with severe fevers and internal hemorrhaging, a condition that would become known as hemorrhagic fever with renal syndrome. By tracing the disease to its source, researchers established a rigid rule for transmission. Human infection occurred through contact with aerosolized urine, feces, or saliva from persistently infected rodent hosts. This ecological isolation led to a foundational hypothesis in virology. Because specific hantaviruses only ever seem to appear in specific rodent species, scientists concluded the viruses and their hosts must have co-speciated, evolving in lockstep over millions of years. Recent genomic sequencing data has moved the virus far beyond the rodent order, decoupling the pathogen from its historical clinical associations. By focusing almost entirely on the viruses that cause human disease, 20th century science missed the true phylogenetic breadth of the entire family. The catalyst for this shift was metagenomic sequencing, the ability to sample and analyze entire swaths of genetic material directly from the environment. The evidence began to mount with the definitive detection of hantavirus RNA in Eulotopatifla, the mammalian order containing shrews and moles. Soon after, virologists discovered distinct hantaviruses in both major suborders of bats, removing the assumption that these pathogens were confined to terrestrial rodents. Then came a significant phylogenetic anomaly. Metagenomic sweeps identified hantavirus-like genetic material in jawless marine vertebrates like hagfish. Today, the known viral footprint extends into multiple orders of both freshwater and saltwater fish, as well as reptiles. Hantaviruses represent an ancient viral architecture that spans multiple vertebrate classes. To understand how the field's perspective evolved, we have to look at the molecular biology that originally placed hantaviruses into the Bunyaviridae family. Early isolates like Hantan and Sinombre revealed enveloped virons containing exactly 3 distinct internal nucleocapsids. Inside those nucleocapsids, the viral RNA features highly conserved complementary nucleotides at the 3' and 5' ends. These lock together to form crucial panhandle-like secondary structures necessary for replication. While this structure matched other recognized Rhinera in the Bunyavirinae family, the hantaviruses lacked a key behavioral trait. They did not rely on an arthropod vector, like a mosquito or a tick, to spread. Without an insect to carry the virus between disparate animal populations, virologists assumed the virus was dependent on the isolated, localized life cycles of terrestrial rodents. Early observations reinforced this. HantaVirus was tightly associated with Apodemus field mice, while Pumalavirus was isolated from bank voles. This combination of unique molecular structure and narrow ecological isolation created an assumption of co-speciation that governed the field for decades. There was, however, one historical outlier. In 1965, researchers isolated Totopalaeum virus, or TPMV, from an Asian house shrew. For 40 years, the field largely dismissed TPMV as a laboratory artifact or a temporary spillover infection from an unidentified rodent host. That dismissal ended in 2007 when field studies confirmed TPMV's natural ecology lay within Eulipotyphlins. As the catalog of viral genomes expanded, researchers applied DMARC analysis, diversity partitioning by hierarchical clustering, to map the genetic architecture. Initially, this phylogenetic tree showed a unified cluster of the traditional rodent-borne hantaviruses. But the DMARC results for TPMV proved it did not fit inside that rodent clade. Instead, it formed a completely distinct sister group. This divergence established that genetic architecture rather than human disease pathology would become the arbiter of classification. By 2017, the Buenaviridae family had become severely crowded. To accommodate the influx of new genomic data, the International Committee on Taxonomy of Viruses promoted the family into the overarching order, Bunyavirales. In parallel, the singular genus HantaVirus was promoted into its own distinct family, HantaVirinae. As this expanding cladogram shows, researchers created the subfamily MontaVirinae to house the explosion of bat, shrew, and rodent viruses, including the classic human pathogens now reclassified under the OrthoHantavirus genus. Mathematically, that requires the HantaVirinae ancestor to date back roughly 300 million years to the late Carboniferous period, branching in tandem with vertebrate evolution from jawless fish onward. There is a flaw in this model. If HantaVirinae strictly co-evolved alongside vertebrates, we should find them in every major animal lineage. Instead, there are unexplained absences across the phylogenetic tree. This points away from co-speciation and toward a more chaotic evolutionary mechanism, host switching. The gaps in the trees suggest that HantaVirinae did not ride quietly alongside vertebrate evolution. It jumped across distinct, highly separated biological lineages. Ecological data increasingly supports this host-switching model. Specific Hantavirica are now being found in multiple distinct host species depending on local geography. The mechanical driver for these jumps is genomic segment reassortment. Because the viral RNA is separated into distinct segments, different viral strains infecting the same cell can swap genetic material, allowing them to adapt to a new host. New metagenomic data extends the viral reach into a new biological domain. Researchers have detected hantavirid-related RNA-directed RNA polymerases inside insect populations. Specifically, these viral signatures were found inside Curcullionid diapreps root weevils and pearlid stoneflies. This has led to a hypothesis now circulating in evolutionary virology. The true origin of these supposedly mammalian viruses might be ectozoan. They may have started in arthropods. The study of hantavirity has evolved into an expansive, unsolved mystery that now reaches deep into the invertebrate world. Future structure-guided designs will focus on obscuring non-essential regions, forcing the immune system to target these conserved pockets and produce truly broad-spectrum protection. The MV Hondias outbreak illustrates the speed of zoonotic emergence. To stay ahead, the pharmaceutical industry must move past reactive strategies.